All right, question number 1. Which statement best describes what happens when a gram positive bacterium is placed in distilled water and penicillin? Well, in order to tackle these questions, we need to know what a gram positive bacterium looks like, what is its structure, what does penicillin do to the bacterium. We need to go back to the options and know what isotonic is, what isotonic is, what osmotic lysis is, and understanding the other options. So first things first is isotonic. What is it? It is the no net movement of water. B, we have water will move into the cell. So we have a picture here, number 2, that water is going into the cell, and that is hypotonic. We have C, water will move out of the cell, and that's the image number 1, and that is hypertonic. D, the cell will undergo osmotic lysis, and that's image number 3. While water is moving into the cell, we have an expansion of the cell, causing the cell to break, to lyse, to weaken the cell, which would cause the contents to come out of the cell. So that's osmotic lysis. So I'll put that here. And our last image, water is leaving the cell, causing the plasma membrane to shrink, and that's plasmolysis. We have E, sucrose will move into the cell from an area of higher concentration to one of lower concentration, and that is facilitated diffusion. Now there are 2 types. We have the facilitated diffusion with proteins called nonspecific transporters, and a facilitated diffusion with proteins called specific transporters. And in this case, we are dealing with specific because it gave us a specific molecule, sucrose, which is a large sugar molecule. So we have specific transporter, proteins. All right, fit that there. So now we go to our question. Question number 1, we have a gram positive. We know that gram positives have thick cell walls. And we know that penicillin destroys cell walls. So if you can picture a thick cell wall, positive, a gram positive, and the penicillin coming to attack the cell walls, this will allow the bacteria to lyse. And which out of these 4 options you see that the cell is lysed? Osmotic lysis. And that's what we have. Water going into the cell, because we have the distilled water. We have the penicillin breaking down the cell walls. So we see the cell wall being broken, the contents are leaving, and water's going in. So the answer here is D. Number 2. Which statement best describes what happens when a gram-negative bacterium is placed in distilled water and in penicillin? So we have a gram-negative Gram negatives have thin cell walls, or thin peptidoglycan with an outer membrane. So thin cell walls. We have the distilled water, and we have penicillin. And we already know what penicillin does. Penicillin destroys the cell wall. Since the cell wall is thin, we can look up here at our images to see which one would best represent what's going on in question number 2. So we have water going into the cell, and it looks like the answer is B. Water will move into the cell. Number 3. Which statement best describes what happens when a gram-positive bacterium is placed in an aqueous solution of lysozyme and 10% sucrose? Well, we know from above that our gram-positive has a thick cell wall, and it's an aqueous solution of lysozymes. We need to know what a lysozyme is. Well, it is an enzyme that breaks down cell wall. So it breaks down cell wall. And we already know that gram-positives, they have a thick cell wall. We also have sucrose, which is a sugar. So what happens in this solution is that the sucrose binds to the protein, the enzyme, creating, so we're going to put binds to the protein to form hydrogen bonds. So the sucrose in the solution kind of like distracts the lysozyme, so the lysozyme will not go and break down the cell wall of the gram-positive bacterium. So with that being said, what would be the answer? And if you chose A, that is correct. Question number 4. Which of the following is not a distinguishing characteristic of prokaryotic cells? In order to answer these type of questions, you need to find out what are the distinguishing characteristics of a prokaryotic cell. So we're going to go down the line. A, they usually have a single circular chromosome. That is true. So we're going to put a P right by it. B, they lack membrane enclosed organelles. That's usually eukaryotic cells. C, they have cell walls containing peptidoglycan. That is so true. D, their DNA is not associated with histones. That is true. That's eukaryotic cells. E, they lack a plasma membrane. That is so false. They have plasma membranes. So the answer to this question is E. Number 5. Which of the following statements best describes what happens to a cell exposed to polymyxins that destroy phospholipids? And what we're going to focus on right here is exposed to polymyxins that destroy phospholipids. Phospholipids are components of plasma membranes. So we're going to put plasma membranes. Plasma membrane. Right here I have a picture of a cell. We have the cell wall on the outside, the plasma membrane, and then we have the components inside of the cell. So what happens when polymyxins attack the cell, well, it attacks and destroys the phospholipids. So we're going to get rid of the plasma membrane. And this is what we have. We have a cell with components and a cell wall, peptidoglycan. So what we see here is that what's inside can come out. The components can come outside of the cell since there's no plasma membrane to hold them in. So that's the picture that you want to hold in your head. Now when you're going through A through E, your answers, your options, you're going to find the one that represents what you're seeing here. A. In an isotonic solution, nothing will happen. Well, this is not an isotonic solution. B. In a hypotonic solution, the cell will lyse. Oh no, the cell did not lyse. It's just the phospholipids were destroyed, but the cell wall is still intact. C. Water will move into the cell. Yeah, water will move in the cell, but you want to be very specific with the answer. D. Intracellular contents will leak from the cell. Since the plasma membrane is no longer there, this will give the opportunity for the contents inside the cell to leave the cell. And the answer is D. Which of the following is false about Fimbriae? You need to know what Fimbriae are. And they're short appendages that are for attachment or adhesion. So we're going to go down the list. A. They are composed of protein. B. They may be used for attachment. That's true. So we're going to put an F. They're made of protein. They're used for attachment. They are found on gram-negative cells. That is true. They are composed of pilin. The last two that we have are they are composed of pilin and they may be used for motility. Well, we already said that it's made for adhesion. It's used for adhesion. So we know that they are composed of pilin. So the answer, they are not used for motility. That is the correct answer. They are not used for motility. They are used for adhesion. 7. Which of the following pairs is mismatch? Glycocalyx for adherence. That is true. B. Pilin for reproduction. Well, pilin is used to transfer DNA from one cell to the other, and it's also used for motility. We have C. Cell wall and toxin. Well, some of the components for the cell are on the cell wall can be used as toxins or be considered as toxins. Cell wall protection. Well, the cell wall protects the bacterium. And E, plasma membrane transports. Well, there are transporter proteins on the within the plasma membrane that allows large molecules or a little smaller molecules to enter inside the cell from higher concentration to lower concentration. So we have true for this one, true for this one, and true for that one. So what we're left is with B. The answer is B, because I said that pili are used to transfer DNA information from one cell to the other, and it's used for motility, not for reproduction. Question 8. Which of the following pairs is mismatched? A. Metachromatic granules store phosphates. That is true. And some of those phosphates are used to synthesize ATP. B. Polysaccharide granules store starch. When coming into contact with iodine, the starch turns blue. So that is true. C. Lipid inclusions. They consist of polybeta hydroxybutyric acid. So that's true. D. Sulfur granules have energy reserves, and that is true. They oxidize sulfur and sulfur-containing components. E. Ribosomes are protein storages. That is false. Ribosomes are the site where proteins are synthesized. So the answer is E. It's mismatched. 9. You have isolated a motile gram-positive cell with no visible nucleus. You can assume this cell has. Pay attention to your questions. Motile, it moves, so it either has a pili or flagella. If they have that, then protein is needed to create the flagella or the pili. It's a gram-positive cell, which means that it's a prokaryotic cell. It's a bacterium. Yeah, let's go back. All right, so you know what you know. Let's go down the list. A. Ribosomes. Well, ribosomes, like I said before, is the site where protein synthesis occurs. So we're going to put a star by that. B. Mitochondria. Well, eukaryotes have mitochondria, so it can't be that one. So we're going to put a false. C. An endoplasmic reticulum. Well, eukaryotes have that, so we're going to put a false. A Golgi complex. Eukaryotes have that. We're going to put a false. And since we have false answers here, or options, E cannot be an answer. So we're going to put a false. So the only one that we have left is A, ribosome. So the answer is A. Our last one. Number 10. The antibiotic amphotericin B disrupts the plasma membranes by combining with sterols. It will affect all of the following cells except. So now we need to know, out of these options, which ones have sterols? Well, eukaryotes have sterols. Animal cells, sterols. Gram-negative bacterial cells, we're going to put a question mark. Fungal cells, mycoplasma cells have sterols. Plant cells, they have sterols. So the only one that does not would be gram-negative bacterial cells. So the answer is B.